The world of molecular diagnostic is in need of faster, cheaper, and more reliable solutions. Artificial intelligence accommodates this requirement by enhancing throughput of gold standard tests such as PCR. We are all familiar with molecular tests as we all have done one for COVID. These tests require a sample collection, the genetic material extraction from the virus, and the recognition of its nature using a technique called PCR. And PCR is capable of detecting the presence of a specific target, but many people consider it as a binary signal, positive or negative, which limits the power of PCR to the detection of only one target at a time. In my view, PCR is more than that. Suppose I want to detect 3 different viruses simultaneously. For this, I will need 3 individual tests, which will result in 3 different signals. However, if I had limited resources, I will need to reduce the cost and the sample that is needed. So I can use a technique called multiplex PCR. And multiplex PCR is capable of detecting many targets at the same time, with the downside that the signal that I will produce will still be a single curve, and I won't be able to tell what virus is amplifying. However, if we look closer at this curve, we can imagine that each of them represents a fingerprint of a certain virus. Therefore, by analyzing them using an artificial intelligence, we can identify the pathogen correctly and provide precise treatment to our patient, optimize the use of the drug in the hospital, and all of this in a cost-effective manner. This is how data-driven multiplexing was invented. This method uses artificial intelligence to analyze the entire PCR curve and recognize different targets in a single well reaction. Here is an example of one of our recent work where we identified the presence of 7 different respiratory infection-related pathogens in a single reaction with nearly 99% of classification accuracy. For the first time, we can recognize the biological nature of a pathogen by only looking at the amplification curve shape resulting from PCR reaction. The past 2 years of pandemic highlighted how heavy the cost of diagnostics are. We need to have a better solutions for multi-class infection so we can have a faster, cheaper, and more reliable diagnostics. With my PhD project, I'm aiming to make diagnostic simpler and available for everyone. Thank you.